Hello guys, a warm welcome to my YouTube channel. Today I'll be discussing about the transmission of nerve impulse and synaptic delay. What is meant by transmission of impulse? In simple words, transmission may be defined as the passage of electric current or impulse from one neuron to other across the synapse. So, in this diagram, you can see the synaptic knob which contains many vesicles. These vesicles are colored in blue, and this is the synaptic cleft, and this is the postsynaptic membrane. Drawn in green are the receptors of the postsynaptic membrane. So now I'll explain the transmission of nerve impulse in detail. So in the previous video I have explained about the conduction of nerve impulse. So let us consider a condition where the axon is conducting an impulse and the impulse has reached the synaptic knob as illustrated by this arrow. When this impulse reaches the synaptic knob, there is opening of calcium channels at the synaptic knob. Numerous calcium channels are opened. So these calcium channels will push the synaptic vesicles. And when the synaptic vesicles fuses with the cell membrane of the synaptic knob, there will be release of chemicals. These chemicals are of two types, excitatory and inhibitory. The main excitatory chemical is acetylcholine and the inhibitory one is dopamine. Now let us see how the excitatory chemical, that is acetylcholine, works. So I have already mentioned that owing to the pushing by the calcium ions, the vesicles are fused at the membrane of the synaptic knob and they release chemical. When acetylcholine is released, it is being acted upon by an enzyme, acetylcholinesterase, which breaks this acetylcholine into acetate and choline. The choline will re-enter the synaptic membrane and is used for the resynthesis of acetylcholine, whereas the acetate will bind to these receptors on the postsynaptic membrane. When this will bind to the receptors on the postsynaptic membrane, this will result the opening of sodium gated channels in the postsynaptic membrane. Numerous sodium channels will be opened. As a result, the second neuron will be stimulated and an action potential is said to be developed in this second neuron. Now let us consider a condition in which inhibitory chemical, that is dopamine, is released at the synaptic cleft. Whenever this inhibitory neuron dopamine is released at the synaptic cleft, this will bind to the postsynaptic membrane receptors. The binding of the dopamine on the postsynaptic receptors will result in the opening of chloride channels. As a result of this chloride channel opening, the postsynaptic membrane will further become more and more negative and will be in hyperpolarized state. Thus, the generation of action potential in the second neuron is inhibited. Now let us understand what is meant by synaptic delay. So as there is the conduction of action potential along the axon, when it reaches the synaptic knob, there will be movement of calcium ion into the synaptic knob, and when this fuses to the cell membrane, it results in the release of neurotransmitters. This entire event, entry of the calcium and then the subsequent release of neurotransmitter into the synaptic cleft, results in some delay of nerve transmission. This delay is quite some milliseconds in nature and is referred to as synaptic delay. So with this I end my video. If you have liked it, do share, like and subscribe. Thank you.